everyone. Can you hear me? Okay.
Yes.
Hi, everyone. I'm Michelle. I'm with GB Flynn's lab, or Origami Lab. Today I'm going to talk about this framework that we have developed called Nipoppy. Okay. So first I'm going to start with a little bit of a humorous thing. It's a comic that we designed, drew, created. And if you have ever been in a situation like that, I would like to know, because we have definitely been in something like that. So say you are a grad student, postdoc, other academic position. You arrive in a lab and are told maybe, hey, we have this data. Could you do something, do this analysis with it, and also do it quickly because we want results and we want to publish. And then you're like, yep, sounds good, mission accepted. And then you're like, oh, the data is in this folder, let's take a look at this folder. And maybe it's not, and then there's like, oh, little shock. It's not the easiest thing to understand. It was maybe collected a long time ago by other people. There's a lot of directories. It's unclear what anything is, and you open a file that's called demographics final, although there's also another file called final demographics, and then there's things, and then it's just very messy in general, and then like, oh no, what did I get myself into? This thing that I thought was going to take 1 month is going to take maybe a lot longer. So here, finally, after, here we said 1 year of sleep deprivation and a new caffeine addiction, everything is done, the paper is submitted, my novel findings with pretty plots, which is everything you would want in a paper. And then again, sometime later, you get some reviewer that's like, could you redo the entire thing, but with this other data set? And then, maybe you're not very happy about that. Okay, so I'm not going to ask anyone I'm not going to ask the whole audience if you've ever been in a situation like that. But some of us have, I think, and this is what we're trying to help with. So in more formal way, for a more formal introduction, the question that I'm trying to address is how can we make neuroimaging data sets more fair? And of course, there's a lot of tools and platforms that we have already seen today, but this is another way. So FAIR, for those of you who don't already know, stands for findable, accessible, interoperable, reusable. It has open science principles that we think are very important. So there's a lot of existing tools and technologies that have been developed by the neuroimaging or broader community. So first we have BIDS, which as established, everyone pretty much already knows about, Aptainer or other container technologies for being able to run a software with its dependencies on any platform. And then finally, this other thing, which you might be less aware about, it's another framework called Boutiques, developed by another lab in Montreal. Actually, it was also mentioned in the previous talks, if you paid attention. So Boutiques is a descriptive framework for describing a command line. What is a command that is run, and what are the parameters that can be used with this command? So it's a way to formalize details of the command line. I'll go into a bit more detail for that later and also show some example. So with Nipoppy, we leveraged existing open science tools to build a flexible framework for organizing data and also running pre-processing in a reproducible way. So one thing that I think is important to stress about Nipoppy is that the goal is, of course, to help facilitate organization and processing of data. But we also think it's very important to have a training aspect, to teach best practices, and also make it easy to implement them. So just like wet labs, for example, have standard operating procedure or protocol for how to do experiments, we think of Nipoppy as also kind of a protocol, but for after you've acquired the data, what do you do with it? So this is, first, there is data capture. We don't really touch that, mostly because a lot of the time, in our experience at least, when you start working on a study, the data has already been acquired. And then you just have to deal with whatever shape it comes in, which sometimes is not very great. But then that's something that you can think of in the future when you do a new study. Maybe think about this a little bit. Spend more than 0 seconds thinking about how you're gonna name your files. So first step after data capture is data curation or organization. So one thing about studies is that, sure, there is neuroimaging data, but also there's also other types of data without which your neuroimaging is not very useful. So if you don't know the participants, demographic information, maybe some assessment things, then you can't do much necessarily with the neuroimaging data. So here we have for the imaging data, of course the bid standard exists, so then that is very easy. We say, put your data into bids because that is best practice, and a lot of people in the community already do that, and then you have interoperability with all of the bids apps and other software. But then we also have what we call the tabular data. So here we think more commonly of demographics, cognitive assessments, clinical assessments, kind of thing. So we also provide some guidelines. They are still a little bit in flux, I would say, and we're still thinking about it because we want to have this be a community effort. And a lot of people do different things with the tabular data, and there's a lot of different ways, and all of them are valid. But we have some guidelines and some things that we think kind of make sense. If you want to talk more about this, I'm happy to, but later. After the neuroimaging data has been put into bids and we want to process the data using both containers for reproducible environments, but also a way to be able to keep track of runtime parameters. And this is where the boutiques framework comes in. And again, I will wait more and later for the demo to show that. But one thing is that we put here you can see that there is separation of different pipeline names and pipeline versions, so that there is some kind of standard or data specification for where to put your derivative data, and also so that you can know what has been run on your data. And for both of these parts, we have some tracking that is being done, and this again is very lightweight. So Nipoppy, well, yeah, it's a Python package. It's a CLI. You install it locally. It has some dependencies on scientific Python packages, but otherwise the rest is just like some TSV files, some JSON files, so that you can just take a look at it, and everything hopefully makes sense. So here you can see that there is a manifest file. So that is something that I will go again a little bit more later. But we say that you should, if you are making a study, you are doing analysis on a data set, and you should be aware which participants and which visits or sessions are in your data set. So this is actually a requirement, so you need to have this file. And this has to be user provided. But then with this file, if we know what is in this file, then we can do tooling to know which participants have been converted to bits successfully, because sometimes you try to do bits conversion and it fails for one participant because maybe the DICOMs are corrupted or something like that. We can also know who has been processed successfully with different pipelines, and I'll show an example of that later. And then once the data has been processed, we also have this extra step of extracting features for downstream analysis. We call this these features, if you adopt UK Biobank terminology, are called IDPs. So that's what we're using here. So you can think of if, for example, you process your data with FMR prep, but then you still have to do some additional steps, and maybe they are more custom for your specific analysis. So you kind of just write a script. Maybe you containerize it. Maybe you don't, although you should be containerizing it. So that's what we call the extraction step. And all of these, that's okay. All of these curation, processing, extraction, all use other pipelines, so bits conversion pipelines, preprocessing pipelines, extraction pipelines also. And the way to run these is all very similar in Nipoppy, so that's what we're trying to help with. And then finally, there's the analysis part, which is where you use your fancy machine learning or awesome statistics comparison kind of things. Okay. This is cut. Sorry about that. But to summarize, we go from raw scanner data to extracted IDPs, and we also have some extra things to visualize tabular data or metadata on a dashboard. Here you can't see the link, but it's called digest.neurobagel.org. It's okay, I think it's on the next slide. Yeah, so there's this website. If you do the previous step, actually here, sorry about that. If you get this processing status file with, that who has been processed successfully with different pipelines, you can upload it to this website, and it will generate some interactive bar charts that you can look at and filter to get the list of participants who have all of your pipelines run successfully. I think it's gonna scroll down in a bit, and then you see this, and it's also divided by session. So that's a quick sanity check. It's not quite a dash. Well, yeah, it is a dashboard actually. It's a dashboard. You can look and then have this dopamine surge if most things are green or not, and be sad about it. The second part of Nipoppy is that it's data organization specification. So as you have probably noticed, we say here you put the bits data, and then here you put the derivatives, and here you put the tabular data. So because, again, similar with BIDS, and actually we are talking with the BIDS community to try to align with BIDS 2.0, future version of BIDS, to have this study level BIDS. So instead of just having neuroimaging and some other kinds of modalities, really have this, neuroimaging, but also the tabular data and things like that. Yeah, and then if we know where the data are, then we can build tools around it, as it has been a recurring theme during this educational course. And then we also provide a software package with tools to help work with this framework locally. One thing to note about Nipoppy is that I think it is particularly well suited for decentralized data management and processing. So again, it's a local package. You install it on a computer. You can do local processing. And actually also, if you're curious, there was a brain hack hackathon a few days ago. We have worked with the Neurodesk team at that time. During that time, and you can install Nipoppy in the Neurodesk environment and then have access to all of the container and good stuff that they provide. But in particular for Nipoppy, one of the very good use cases we have found for it is that in consortium type settings where there is possibly a core team and then some sites, and then you really want to make sure that all of the sites do the processing in the same way. But maybe they have different technical expertise and also different levels of computational resources. But we can still have this teaching process and have independent, because they don't share the data between the sites, but consistent processing, so that when you share the IDPs, you can be reasonably sure that that everyone did the same thing and that it makes sense to actually pull the data together. So here's just a little graphic that I made, where you can have different sites, and then all of them use Nipoppy, and then you don't have to interact between the data sets. And then finally for this also, we don't write processing pipelines or bits conversion pipelines. So it's really like whatever workflow that you have that you are already doing, maybe you could encapsulate it and then run your analysis with Nipoppy. And then if you have multiple pipelines, you can also make it so that you can run the pipelines in the same way, which I will show later. Okay. So for typical Nipoppy workflow, so hopefully that will make it a little bit clearer what I'm talking about. Say you have some raw imaging data in DICOM or in Nifty format. You can run Nipoppy in it. It will create this empty directory structure with different directories where you're supposed to put the different types of data. And then there are two user-provided files. Actually, it's really more one user-provided file. It's this manifest.tsv, which I have already mentioned. It's supposed to be a ground truth list of who is supposed to exist in the study, so that you can know, for example, you recruit a new participant, then you update the manifest, so that when you have the data, then you can run through all of the bits conversion and then the processing, etc. And if you know bits, then you might notice that this is kind of similar to the participants.tsv, but the manifest is really a requirement. yeah, the other file is the global config, which I think I will skip for now, but it's yeah, I'll skip it for now and talk about it more during the demo. And then you can install processing pipelines. And what does that mean? So most processing pipelines probably are containerized, so that involves downloading the container, but also some lightweight JSON files that have information about runtime parameters. And yeah, so here we have, for example, some bits conversion pipelines, some versions of FMR prep in case you wanted to compare different versions. Okay. There is some reorganization step for preparing for the bits conversion. And then there's Nipoppy Bitsify, which is convert the data into the bit standard. Here is an example of what this is part of the boutiques framework. It's called an invocation file. So you can specify runtime parameters, and here it's kind of those special expressions because Nipoppy will come in during program runtime and will replace those by actual paths because it's aware of the paths. Where the bits data is supposed to be, where the source data is supposed to be, and then which participant is being converted at that time. Similar thing for running the processing. Again, I will show that a little bit in more detail during the demo. Here. For tracking the data, there's Nipoppy track processing, and it will generate files like this. And again, the idea behind this is very simple. Basically, as a user, you define, and there are some defaults, but you can define it. For a pipeline to have completed successfully, which output files do I expect for every participant and maybe session? And then it's just a list of paths, and then the tools just go and see for every participant, do they have all of these files existing? If yes, success, if no, fail, and some other things. And then finally, Nipoppy extract. Here's just an example of some tabular form IDPs that can be used in an analysis downstream fairly easily. Okay, so briefly, takeaways before I go to the demo. Nipoppy, again, is a framework that combines existing tools. It provides guidelines for data management. It can help with reproducible MRI processing and IDP extraction. And it has some nice features like progress tracking, which is useful for when you have more than one participant. Okay. For analytical flexibility specifically, Nipoppy can make it easy, similar to previous tools, actually, to run different pipelines in different versions. And it stores them in independent, parallel kind of directories. And then also the runtime parameters are stored in JSON files. So yeah, here's an example, actually, of something that we did recently in our lab. There's a postdoc, Brent, in our lab. He ran track-to-flow, and also, which is a pre-processing pipeline for diffusion data, and also QSI prep, which is also a processing pipeline for diffusion data, and then compared free water estimates for different ROIs. So here's So before I go to the demo, I just have a thank you slide because this is a team effort. So here's parts of the lab, mostly people who work closely on Nipoppy and some other related projects. Okay. So now if you could switch to the laptop. Okay. I think I will have to. Okay. I think I might increase this a little bit more. Is this okay? Can everyone see? It's gone again. Okay.
If there are any questions, invite, please, use the time. Analytical, technical puzzle. Otherwise, just rest. Yes.
Yes?
Could you explain a bit the differences in the goals between Nipoppy and Babs, if you're familiar with Babs, or maybe the Babs people can comment as well?
Yeah. Well, I see Satra's also here, so maybe he can add on after I say something about that. I think they're fairly, so part of Nipoppy is the processing, running processing pipelines. That part is similar to what Babs does. Babs is a bit more focused on running on HPCs and on implemented this fairly big framework with all of the data lab things. So then with Babs, you can get in the Git logs, information about how you ran the pipeline. Nipoppy is, part of it is, one component of it is the pre-processing, but really as a bigger whole, there's also like the bits conversion part, and then what happens after the pre-processing, and the data specification aspect of it. So it's more of an end-to-end thing, I would say.
And so I think Michelle covered that pretty well. The key with Babs, I think, is leveraging data lab and making that go under the hood in many ways, because working with data lab is not the easiest thing on the planet. There's a fairly steep learning curve that goes with it, and this was an effort to put a layer on top of it, but still use those toolkits to track versions consistently. Because there are a lot of advantages of a framework like data lab. It's just a complex framework.
Okay.
And I guess to some extent, it also targets a little bit slightly different communities as well. I mean, if you have to use data lab, then it does imply some kind of a fairly good technical background in some ways. If you don't, there's that technicality is less. Technical aspects are less demanding. So there's the scope aspect, which is a bit different, as Michelle and Sandra said, and there's also the who is more targeted as a community by what tools. I mean, those things are, and I guess we've seen that for all the tools actually. I think BrainLife in particular seems to be very end user and very GUI, sort of centric, and I think that's also slightly different communities. Like we had a question from a clinical person that seems to be very interested by BrainLife because of that easiness of things. But then maybe there's more researcher communities that would be appealed by like a Neurodesk or others, framework, Nipoppy or others. So it's not only it's hard when you don't know intimately all those tools that that they seem to be all doing kind of from the distance similar things. As soon as you kind of zoom in, there are sufficient differences that it's all good that they exist. They coexist together. No luck with
No, they said.
Okay, so we'll skip
no,
the. I guess the.
yeah.
It's
okay. If you have more questions, or actually we have time for questions now, or does anyone have more questions? If you want a demo on my laptop later today, I am happy to give one.
Is it possible to run on Stefan's laptop?
I just need a terminal.
I can ask Stefan.
Actually, we might have a backup computer possible, since we have prior evidence that the connection. We can try. I can connect to a server. Yeah. I need a terminal.
She just need a terminal.
I can.
Okay, let's remove that.
But it doesn't have to be. Actually, it might be.
There's no break in between. Yeah, you're gonna come up next after Michelle.
Yep.
You can take a quick drink. We're gonna try her demo. Yeah. You can take a seat first.
Oh, okay. Is this big enough for everyone? Yes, okay. All right, let's do this. This is not where I prepared the demo, although I do have things there. Nipoppy demo. All right, so here I have a bits data set. So we're gonna start with the bits data set, so because it's gonna be quicker for demo purposes. I'm gonna move this thing to backup for now. Make a new directory called demo study. So here I am gonna activate actually first a Python environment with the Nipoppy Python package installed. So I have available to me the Nipoppy CLI. If you do -h, it's gonna print out some information like this. It looks like this. And I move into this demo study. Currently it is empty, and I can do similar to git init, or also babs init actually, and then say that I want to use source data ds4. Yeah. So this is what's downloaded from OpenNeuro, and also modified, so it's a bit smaller because I don't need everything for the purpose of that. And it's gonna might take some time. Okay, usually it's a bit faster than that. I'm on the login thing, wait. Taki. Okay. Yes. Oh my god. I can't type today. Activate. Scratch. Nipoppy demo. Demo study. Okay, might be faster now. So here you can see I don't have a file explorer given extenuating circumstances, but those are the directories that it creates. Is also if you already have a bits data set, it will create the manifest file for you. So here you can see I have, I think, 9 participants with only 1 session, and they all have anatomical data. Okay, the next step, if you remember the slide, is to install pipelines. So first I want to see what pipelines are, or I want to run 2 versions of FMR prep is what I was thinking for this demo. So I can do pipeline search FMR prep. It's going to search the Zenodo data repository where we have currently all of the supported pipelines uploaded. And because it's on Zenodo, anyone can upload on it. So you can use the Nipoppy CLI once you have some pipeline configurations ready, it's tested, and then you can just do upload to Zenodo, and it's gonna work. So here I have 2 versions. I think that might, actually, probably gonna work. Okay, there's also, before that, the global config, because the system that I am on does not uses an old version of Apptainer when it was still called Singularity. Okay. So I'm gonna do that, and then Nipoppy pipeline install. So if you go on Zenodo and then you do zenodo.org/records and then put this ID, you will find the JSON files that are associated with that. So here. And it's gonna download things, and I think it should be quick because it doesn't require on. Okay, yeah, we're not gonna download the container because that's gonna take a bit longer. So I have 20.2.7, and also let's do 24.1.1. So again, we are thinking of these tools as being community contributed, and we're gonna start working on adding the NeuroDesk, some of the NeuroDesk containers to it. Again, no for this. And you notice that here it says we added 2 variables to the global config file, and you must update it, or it's not gonna run. So again, we go back to this global config, which stores basically, the global config should be for users, like the only thing that you need to modify for things to be able to run, for pipelines to be able to run. Because we have, all of the pipelines have some default configuration. If you want to go deeper and modify those default and run like your very specific call to FMR prep, then you can do that. And I'll just show where to do this. Okay. So where did I put my free surfer license file? So here it asked for a path to the free surfer license because you need that to run FMR prep. So I will just copy that so that I have less chances of making typos. And go here again. And that was free surfer license.txt. And then the one below that, you also should specify a directory for template flow. Then, actually, no. We're just gonna copy the same thing here. Like this. Okay. All right. Now we have updated the global config. I said I was gonna show the boutiques things. So during the install step, what it did not download the container, but it can. But we already have the containers downloaded somewhere, so I'm also just going to copy them over. Yeah. Data. Store. So while this is running, I can show, if you go inside the pipelines directory, you can see there's 3 pipeline types, which I have mentioned before. If you go inside processing, which is what FMR prep counts as, and there are, yes, FMR prep actual directory name, there are some JSON config files, and that is everything that's needed to be able to run a container with Nipoppy. So for boutiques, I don't know how much time we have actually. Can someone tell me? Five minutes? Okay, cool. So for boutiques, there is this descriptor file, which I invite you to look into a little bit more. I will just briefly show. So, oops. Okay, that was not a good idea. So there is this command line thing, which looks like a little bit overwhelming, but if you know FMR prep, you know that it has a lot of parameters, and this is basically what this is showing. And then for every input, or for every one of these things, it has some description and some definition of this is not optional, or this is optional, this is a positional argument, or this is a flag, or this is an option. And because of this, then we know how to build a final command line and how to run FMR prep. And then there's this descriptor file. Usually it would be provided by the person developing the pipeline because they are the ones who know this information. And then we have, for the user, this invocation file. So for every one of those inputs in the previous file, we can specify a value. So again, we have some special expressions for paths, but otherwise you can also specify things like processing. So these are all FMR prep arguments for different pipelines. You will see different things. There's the free surfer license and some other things, output spaces, which was also mentioned in one of the previous talks. Okay, my containers have finished downloading, so now I can do Nipoppy process and then pipeline with the double dash FMR prep. And then I specify the version because I have 2 versions of FMR prep install. 20.2.7, and let's say I want the first participant, which I forgot what was the ID of that participant. this. Okay. Here. And also I'm not actually going to run FMR prep because that will make us go over the 5 minutes remaining time for this. So then I do simulate. So what it's going to do is instead of running, and it's helpful like that, instead of actually running the command, it's just going to print out what command line expression it has built. So it's this. You can see because of container things, we bind a lot of paths. It is possibly a little bit overkill. And also now with tools like NeuroDesk, you can run things without having to have the extra like singularity, things like that. And that's also possible, or soon to be possible with Nipoppy, where you can just not have the container things if you're, anyway. You can, there's some things that are printed out. They are not always necessary if you run things through NeuroDesk, for example. But then after that, you have the biz data directory, you have the output, you have participants. So this is just a regular FMR prep call with the pass and everything done for you. And then you can do the same thing to run the different version. You just go here and modify the version, which I'm not going to do. And then the last thing I wanted to show is I have yesterday night run FMR prep on this participant. So I think it's in data. Nope. No, it's not in data, it's in backup because I didn't move it there. Backup, derivatives. Yeah, I can just do this. Delete the derivatives directory. And then move backup derivatives here. Okay, now we have these derivatives. And then I'm going to just show the tracking features. So what I'm going to track, again, in that pipeline configuration directory, if you do pipeline processing, FMR prep, let's say 20, and there's a tracker.json. So here is this thing of we have, again, it's populated with defaults. If you have different requirements, you can change them. So basically, this is the list of files that the tracking command is going to check. I can do Nipoppy track processing, and then pipeline fMRI prep, pipeline version 20.2.7. And then it's going to run. And then we also, I forgot to show this because I don't have my list of things to do during the demo. There's a Nipoppy status command. It just creates a summary of the data set. So this is actually very useful, especially as like a sanity check to see. So here, oh yeah, okay. In my manifest, I have 9 participants for this session. In the bits data, I also have 9 participants. These are empty because we didn't do these steps because we already had the bits data. For my 2 pipelines, which I already copied the derivatives from, which is why this one is there, even though I did not run the Nipoppy track processing command yet. It says we have 1 out of 9, presumably. Yeah. And then this file, the tracking file creates this TSV. The tracking command creates this TSV, which looks like this. And then this you can use with other tools, including NeuroBagel, which was not mentioned during this course, but if you're interested, we can talk later. Or this dashboard, online dashboard at digest.neurobagel.org. So thanks everyone, thanks for your patience. I'm sorry about the technical difficulties.